Good morning. Let's get started. Well, first of all, I'd like to thank you guys for coming along. I think I realized there is multiple sessions going on, so I appreciate you guys being here. It's a pleasure to be actually back to this meeting, and it's a real privilege to be able to give you an update of what we've been up to as a company. As you can see from the first slide, I'm going to be focusing, and we as a company are focusing on iPSC-based cell therapies. Now, it's fair to say that for that modality, it's not exactly easy times. I think overall, there's a tremendous amount of progress in the field. There is a potential product coming onto the market in Japan probably next year for heart repair. There is clinical trials in phase 3 trials starting in diabetes and in Parkinson's disease. A larger number than ever of clinical trials starting, yet there are also a lot of companies, especially big companies, stepping out of this field, and to a certain extent that's driven by a number of the hurdles that are still present. I'm going to be talking about that. So I'm going to be talking about two areas that we focus on. I think we continue to believe in this modality, and step by step we and others remove especially some of the technical hurdles. And each step and each hurdle that comes down brings us closer to the vision why we're pursuing this modality, which is to bring curative treatments to larger patient numbers. And so I'll be talking about two areas I just mentioned. One is a bit more established in diabetes, and the other one is a bit a more exploratory newer area in the platform. Focusing on macrophages, and I will end with talking a little bit about the company as such. So if we dive right in and start on diabetes, I think this is, in our view, one of the areas where stem cell-based therapies to deliver islet replacement therapy has a huge potential, both commercially for companies, but also for patients from a quality of life perspective to prevent long-term complications. And I think it's been a long way. As I mentioned, there is really cool clinical data now emerging that shows that the preclinical data actually does translate into humans, and that gives us even more encouragement to continue the path. We've been at this for quite a while, and we're starting to tick some of the key boxes. So one of the main hurdles, as we hear literally in every talk, and that's certainly also the case for iPSC-based therapies, is manufacturing. We're now able to manufacture in 3D in a scalable way from a GMP line, what we call islet-derived clusters, or ILCs. We have amazing preclinical data, and I'll be showing some of that to you in a minute. However, we and others are already working on the next generation of potential therapies, which is gene-edited iPSC-derived islet-like clusters. And essentially, this serves the purpose that is described further down on the slide, and the approach that we're taking, which is a two-pronged approach. We're working on unedited cells in a partnership with a company called Sernova, and I'll go into that in a little bit more detail in a second. But this will require immunosuppression, and on the long run, this will not be acceptable to patients, at least not to larger patient populations. And so we are already focused on the next generation therapy, which is gene-edited cells, which what we hope won't require immunosuppression any longer. And as I mentioned, the unedited cells is in partnership with Sernova, and the edited cell project is an internal project, and we're getting close to the GMP stage, and we're looking for partners to help us push this further towards the clinic. So where we are and what's the aim of the collaboration with Sernova is really in combining iPSC-derived cells with the really cool medical device that Sernova has developed. It's called Cell Pouch, and they have really great clinical data using cadaveric islets, so not iPSC-derived, but cadaveric islets, on demonstrating that the pouch provides a great environment for islets. Islets are sensitive, they need a lot of oxygen, and are prone to actually die if the environment is not perfect. And so they've really convincing clinical data. They've shown the device is vascularized, that this is what they call can actually be a biohybrid organ. And we have been working together for quite a while, and we have demonstrated that the cells and in combination with the device, preclinical mimics the clinical result that Sernova has. So this combination of cells that can be manufactured at a large scale in combination with a clinically validated device is actually really cool. And you can see at the bottom left just 2 images from an animal study where we've implanted iPSC-derived cells in the device in a mouse model. And on the left-hand side, the green stain after, I think it's a 6-month study, is actually insulin staining, so the cells are perfectly healthy. On the right-hand side, it's actually the red staining, that's insulin, and the green staining is actually blood vessels. So you can see there's blood vessels growing in. If they weren't, the cells would have died. So what does this look like from a clinical supply perspective? And that's shown on the right-hand cartoon. It starts, of course, with GMP manufacturing. As I mentioned, we're at a scale that's appropriate for early clinical study, at a low liter scale, but the format that we're using, it's partly automized. It's in a bioreactor platform we believe is scalable. And so we believe it's absolutely realistic to actually move to a scale where we can produce 50 or 100 doses in one go, which is not what we can today, but what we can do today is sufficient for the first clinical trial. So the cells are manufactured, they're frozen, they're shipped, they're finalized, and then they're shipped in a controlled environment to the clinical site. Now it's fair to say that this is only the first step, but before I get to that, I just want to show you some of the data. And the data on the left-hand side, for those of you who are not familiar with diabetes preclinical model, essentially you take a mouse model, you make the mice really diabetic by killing the pancreas, you transplant the cells take a bit to mature, but you can see eventually the blood glucose gets completely normalized. And this normalization we've now tested for significantly longer than a year. And this is really what gets us out of bed. If we can translate this type of sustained action into humans, and there's currently no indications that the cell start losing their function, this is just amazing. And on the right-hand side, there's just different data that demonstrate that C-peptide levels go up, which is an indication that insulin is being produced. The C-peptide level on the top right of the transplanted cells in the device are very comparable to actually primary donor or cadaveric islets. So the cells that we're making, they have equal potency, and it's just amazing to see that this works that way. At the bottom is just a measure of glucose tolerance that illustrates that there is no overshooting or the danger of hypoglycemic events. So now this is all great, but as I just mentioned, this is just the first step. I think the best-in-class therapy, and that's feedback from patient groups, will require a very mild or ideally no immunosuppression being present in the treatment. And so we and others have been working on immune shielding concepts based on genetic modifications, and we've actually come a long way. We have now developed a system that not only protects the cells against specific immune cell types, shown on the right, T cells and K cells, but that also provides actually like a tolerogenic microenvironment that hopefully protects the cells from rejection. And we can actually make cells islets from gene-edited cells. This has taken us years to do, and we've completed in vitro proof of concept with human-derived blood cells, and we're in the process of initiating in vivo proof of concept. And the advantage for patients, and we hope this will enable and open up a larger market for this treatment, is really that no immunosuppression is required. Now, it's fair to say this sounds actually like quite an easy task. Any postdoc in any lab can make genetic changes to iPS cells, but it's actually be a year-long journey to identify the right edits, to be able to prove their function, to be able to develop a method that's GMP-able, so especially genetic control and genetic stability and integrity after gene editing is something the regulators pay a lot of attention to, and we've developed a process that allows us to do that, and we've discussed that with the regulators. But then also to develop a method that allows the engineering itself to be done in a very simple way. If you've got a 5-step procedure, it's never going to work under GMP-like conditions. And so I think we're in a really good place, and we're now looking for a partner to push that into GMP and into formal preclinical testing. Looking at macrophages, that's a completely different ballgame. This is a lot more exploratory and more early, but macrophages are actually a very interesting cell type, and we believe they're really cool cell type to treat inflammatory diseases. They have a natural tendency to go where inflammation is. They can resolve inflammatory local reactions. They can phagocytose. They can be engineered. And there is emerging data actually preclinically, but also clinically, that macrophages, at least in an autologous setting, can have beneficial effects in liver disease, in cirrhosis, as shown on the right-hand side, but also at the bottom, they can resolve fibrotic lesions, as shown by charisma. And what we really like about this, I think when we started moving into the iPSC space, I think we had a certain naivety on, we're going to work on all those different cell types. We're going to make T cells and macrophages and beta cells. And as the field has found out, this is really a huge effort and requires big budget as well. So we try to focus on cell types that constitute a platform, and they can actually take us into a whole range of different disease areas. And macrophages is actually one of such a cell type, and it's just illustrated here. So we currently, our program focus on fibrosis in liver and lung, because that's where also macrophages naturally go if you give them systemically. But there are a whole other range of indications that can potentially considered, and at least one of them, life-threatening lung infections, is something that we're looking at internally. But we know we can actually make them, we can manufacture them in 3D, we can freeze them, we can make them at a scale that is compatible with first clinical testing. We can genetically engineer them to express targeting moieties, which is no simple task in macrophages. Actually, this has taken us quite some time. We have proprietary targeting moieties, and I think we've actually created first data speaking to the cartoon on the right-hand side, that macrophages help to resolve collagen lesions, and that they can actually phagocytose myofibroblasts. There's quite a lot of more work to be done on the disease-relevant side. So we have actually built 3D organoid models that mimic fibrosis, liver fibrosis, and I think these are the things that we're currently pursuing to then identify partners that help us push this forward. And so just a few words on our strategy and how we work in general. So I think over the last 10 years or so, we've built an integrated platform to take cell therapy projects from initiation into the clinic. I think we have a certain focus on stem cell-based therapies. You can see the main sites are either in Germany or in Italy. I think we also, on the very right-hand side, some of the preclinical capabilities and clinical services. We rely on the larger Evotec global capabilities, but it allows us to take projects actually all the way. And you can see in this middle part what I've been describing here, we invest in early innovation, in novel technologies, create project starting point, and then look for partners to help us push them into the clinic. And the way we work with partners is very partner dependent. But in addition to that, and this is illustrated at the bottom, we also offer services individual stages of the value chain. We license out some of the technologies that we've developed. A lot of that is iPSC-based, but especially in the manufacturing side, we actually also are active in the autologous space and act as a CDMO. And if you look at the last slide, this illustrates the overall activities. We have 2 partnerships ongoing. One I mentioned with Sernova, one with Novo that is just focused on developing technologies for manufacturing of stem cell-based products. We have a number of partnering opportunities that I've mentioned to you on diabetes and macrophages, a couple of others. And at the bottom, I've just outlined the services that we provide. Thank you for listening.
Thank you very much, Evotec. We now have